Toxoplasma is probably the most successful parasite in the whole world. So that means that every time I say that and there's 100 people in an auditorium, say about a third of you here have the parasite.
And everybody looks at the people on either side and say, one of you has toxics, it's not me.
Yes. Or some of them just like look at themselves like me.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Remember learning about ATP synthase in biology and how complicated it seemed? Toxoplasma gondii says, hold my beer.
Our guest today is Dr. Diego Huet, faculty member in the Department of Pharmaceutical and Biomedical Sciences in the College of Pharmacy. Diego, welcome to the podcast.
Thank you. It's great to be here.
So the question we love starting out with is to have our guests tell us about their pathway to science, because we know where you are now, but when was it you felt you wanted to do something in science? Were you in kindergarten playing with your chemistry set or?
Yeah, well, not with my chemistry set, but with bugs. I grew up always with this. I think my dad and my mom really gave me a lot of love for nature, and I was always interested in bugs, but not other animals. I don't really like farm animals. They're too common, they're too big. I don't really know how to read them. And so the bugs provided me with another venue for that. And my parents encouraged me. They bought me all these encyclopedias. And so back at my parents, I have all these giant encyclopedias that were also not for kids. There was I have one where you have all this phylogeny of beetles with scientific terms that I would just look at the pictures. I will not know what this is about, but my dad will just find them in libraries. Like, hey, another bug book for you. So,
did your parents, were they scientists that kind of studied those things? Or
not at all.
Wow.
So my mom was secretary, and my dad was he retired, a French professor, so a teacher.
Yeah.
So and most of my family are musicians. I'm the only scientist.
Interesting.
The
Same,
yeah.
I have to say. But that's nice that they encouraged that early on. So then when did you decide to take it further for a career?
It started out in high school when I really like science, physics, and not really maths, but more biology. And I like the messiness of biology. I think math was too exact for me, and I was good, and then at some point it started to get worse, and then I just dropped it completely. So I started to focus more on biology. So in high school, I was really interested. Also, that was a time when, back in the days when Jurassic Park was at the movie theater. So it was like, oh my God, dinosaurs and genetic modifications. It's like, that's something that I want to really do. Starting with that, I was just really enjoying biology in general biology, and then genetics and so on. And when I started in college, I was in France for my studies. So there's no minors or majors. It's just the cell biology branch. And so I just took this class, and that's what I really got. I fell in love with biology.
So did you actually have a parasitology class as an undergrad?
Not a single one.
Okay.
So yeah, I was first interested in developmental biology.
Yeah.
I thought it was fascinating how you start with one single cell to a whole organism, coming from a single cell, and then you can have body plans like as complicated as of us, of course, but also shrimps or crabs and so on. And then I realized I had to kill a lot of animals for that. So that's where I was not really keen into doing that.
Yeah. So in the French schooling system, can we talk about that for a second?
Yeah, of
because
course.
It's very different from the United States. You have to kind of decide pretty early on what path you're going to go on, right? So you have to say I'm interested in science.
Definitely.
Yeah.
So even in high school, you have these 3 branches. They have letters. So it's S as in science, ES as sciences économiques, so economical sciences, and L as literary. So right there, you already start branching out into, if you want to be more into arts, and if you want to be more into social sciences and economics, or science. And science branches out more, each one of those branches even more, but in the science one, also you can do maths and physics, or chemistry, and also biology. So you have all these different paths. That's just right off in high school, right?
Right.
So,
yeah, that's probably amazing for a lot of people to think about who are educated in the States, because it's kind of like you can kind of be undeclared, for a long period of time. And people try to say that you need to know all these different things. But it's almost like if you want to do this.
Yeah, I mean, for me, it worked out because I really wanted to do that. But I can see that I have a friend who went to the arts section and he was really also interested in biology, but he was not getting good grades on any of those things. But now he's in Australia doing environmental sciences. So it worked out for him. It was just high school was not a good experience for him.
So then in the same system, you kind of just do college the same pretty much as the US, and then your graduate studies, did you do that abroad as well?
Yes. So I'm half French, half Mexican. I was in a French school in Mexico City. So that's where I did my high school. And after that, I moved to France, where I did my undergrad. And in the French system, the undergrad it's only 3 years, so instead of 4. And then I wanted to do a PhD, but to do a PhD, you need to do a master's first. They don't accept you right away from the undergrad. So I did it's like the undergrad it was molecular and cellular biology. And then after that, a master's, and the master's is more specialized. It's also the class size is smaller. It was only 20 of us in the masters, where the undergrads like in the US is 90 people. And in the masters is when you start having more hands-on experiences, where you ask for internships. And there's a lot of laboratories around Paris when I did my masters, where basically they list all these universities that accept students for an internship in masters, because there's no rotations. It's not like the American system. You do 6 months internships in a lab, and that's whether you like it or not. So if you don't like that lab, you just quit, and then you need to get credit hours somewhere else. Luckily enough for me, I was looking still into those. I did a 6 months internship in a lab on developmental biology, working on neural crest cells. And how they migrate through the body. And that's what I realized, that I had to sack a lot of female mice to get the embryos, because I needed to then use the microtome to section and then see where the cells were migrating. And they had mutant mice strains. And after that, after realizing that I want to keep sacking animals, there was this internship offered at the Pasteur Institute. And this is where the person that will become my PhD advisor, Philippe Bastin. And I chose that lab because they were working on these weird organisms, a parasite called Trypanosoa brucei, but they also had tsetse flies. So tsetse flies are the vector of this parasite. And I was like, oh my God, they have bugs. That is amazing. I don't know. Because normally when you see bugs in science, generally it's like, fruit flies. And
right.
Yeah.
I mean, they can be exciting, don't get me wrong, but they were not the bug that I was looking for. So when I met with him, he was overly enthusiastic, and I really liked that. And he was very excited about his research. And I think I am a little bit like him. Now I can convey my science in a very, for the best of the worst, with a lot of excitement. He offered me the position to do another 6 months internship in his lab. And once you do that, at the end of that experience, you can decide to continue in that lab to do a PhD. But it has to also the labs don't have money for you. The money comes from, it's called doctoral schools. They have a pot of money, but that pot of money is very selective and exclusive. So they give it to only the top students. So you need to have good grades in order to be accepted in a PhD program.
So apparently you did.
I did those hurdles. I was like, not to brag, I was third on my master's. So there were two people that were like, and I'm still friends with 10 of them. We actually saw each other last October.
Oh, that's great.
Oh, that's fun.
That is great.
That's fun.
You like trypanosomes because they have insect vectors.
Yes.
But then why did you transfer to Toxoplasma, which lacks an insect vector?
So the lab of Philippe Bastin back in the days, so we had another parasitology lab. We were on the first floor and they were on the second floor. And that lab is the lab of Arthur Scherf. He retired recently. So we were plasmodium. And we were sharing the same tissue culture room. So I could see all their pain of getting transfections and having things work. And trypanosomes, when you look at them on a practical side, they're basically like eukaryotic bacteria. They don't need any host cells. You just need to put them in media and they will divide. So I could just basically grab my flask, shake them, and be like, I can pass them tomorrow, and that's it. So they will hate me for that. But at the same time, I was friends with a lot of them. So I became like an honorary member of that lab. So I will be going to the lab retreats. I'll have lunch with them. And Arthur became a mentor for me also, like a co-mentor. Not really an official mentor, but a very good mentor. And so after working with trypanosomes, which I really like, I wanted to keep working on parasites. And I really like parasites because to me they remind me of bugs, of that divergent biology. There's things that we don't know about them. They're weird, quote unquote, right? And I was like, I really love that aspect of biology because there's so much things to discover, so many things to discover. And I asked Arthur, so what parasites should I work on next? I really want to keep working on these parasites. And there was one lab working on cruci, but I was not really keen on cruci because it was still a trypanosome. Everybody was telling me it's better if you branch out of your comfort zone, even though the parasites are not very similar per se. And so I asked Arthur, what do I do? It's like, well, you want to have fun, work with Toxoplasma. But if you want to, maybe suffer but publish something way more significant, work on Plasmodium. And so I decided to work on Toxoplasma.
Why not have fun, right?
So yeah, that's how I ended up with Toxoplasma.
Okay.
So I'm sure we're going to get into the biology of what you study in toxo, but before we do so, maybe we can talk a little bit about the importance of toxo in terms of human health. So in terms of like what percentage of the population is infected with toxo, how people get toxo, and just the basics, I guess.
Yeah, so Toxoplasma is probably the most successful parasite in the whole world. It infects, the estimates vary, but it infects about a quarter to a third of the global population. So that means that every time I say that and there's 100 people in an auditorium, say about a third of you here have the parasite.
And everybody looks at the people on either side
Exactly.
and say, one of you has
Yes.
toxops, it's not me.
Yes. Or some of them just like look at themselves like me. Like,
Or maybe they say that's my brain fog. Now I have an explanation for that.
And it's extremely successful because it gets transmitted via cat feces or also raw or undercooked meat. And so there's certain places in the world. The distribution also varies depending on, it's also cultural, depending if whether or not the country eats raw meat. So for example, France, I'm still toxo negative. I get tested every year out of curiosity. I grew up with cats and I'm still negative. But in France, people love steak tartare. So this is like a patty. I don't like it because it's raw meat, so I'm not used to. And I grew up in Mexico where I was told all my life to eat meat that is very well cooked. Not because of Toxoplasma, because another parasite. So cysticercosis.
Okay.
So when I saw raw meat, I was like, no, thank you. And I tried it, never really liked it. But in France, the Toxoplasma prevalence is extremely high because of that.
Isn't it something like 70%?
I think it's in the 80s. Yeah.
Oh,
Okay.
so in
70,
France, it's a
80%.
much higher percentage.
Yes. It's extremely high.
Interesting. Yeah.
And it's so high that if you are pregnant, if you get pregnant, they will test you right away. Because going back to the parasite itself, a third of the world population has it, but that doesn't mean that you'll develop any disease because the parasite, basically, once it gets in your body, your immune system will put pressure on it and it will go dormant somewhere in your body, generally a place where there's not a lot of immune surveillance. So it could be a muscle or the brain, and it will stay dormant for the rest of your life if your immune system keeps being functional. And it's only when you become immunosuppressed, either through chemotherapy, through an organ transplant, through AIDS, that you will have some symptoms. And then regarding pregnant women, the parasite likes to be in places where there's not immune surveillance and the fetus doesn't have an immune system yet. So the parasite will go and instead of replicating and causing damage in the mother, it will go to the fetus and cause stillbirths, congenital malformations, and things like that.
So we have the basics down. You work on Toxoplasma. As you said, the world's most successful parasite. It's a eukaryote. But what is the focus of your current research with Toxo? What are you looking at?
The focus of my current research is trying to understand more about the ATP synthase of this parasite. So Toxoplasma, like any other eukaryote, has, and most eukaryotes, because some of them don't have it, but has an ATP synthase. And we all know the ATP synthase is the mitochondrion. Mitochondrion is the powerhouse of the cell. So the ATP synthase is the source of ATP in the mitochondrion. In all the organisms studied to date, this is a complex, and you can find YouTube videos about it. It's a molecular nanomotor. It's such a intricate machinery that people have say, oh, this is why an intelligent design exists. Look at this beauty. And you can see it rotating. The rotation causes ATP to be made from ADP and inorganic phosphate. And in most organisms that we are studying, so I'm not studying them, but other people study mortal organisms like yeast, mammals, fruit flies, the ATP synthase has about 17 subunits, 17 to 18 subunits.
Which makes it a really complicated structure.
Already. Yeah.
It's already a complicated structure. There's people are still trying to figure out how it gets assembled. There's models. There's a lot of work being done. There was a Nobel Prize for it. Johnny Walker, I don't remember when he got the Nobel Prize, but he got a Nobel Prize for the discovery of the ATP synthase. And so, in yeast and mammals, they have 17 subunits. And then when you look at other organisms, specifically Toxoplasma, it has 32 subunits. So,
which is crazy.
It has, yes, it's really crazy, and we don't know why. We don't know why it has that many subunits. Because when you look at the very details, the rotor part, the part that actually makes the ATP synthase, the ATP itself, is well conserved. So those subunits, the catalytic subunits that enable the transformation of ADP and inorganic phosphate into ATP, are very conserved throughout evolution, which makes sense because you want them to be, these are a pretty crucial function that you don't want to mess up with that. But then the rest of the enzyme is a playground for evolution, I guess, I suppose. And so you have all these different subunits that are completely different. Some of them are different just by sequence. So the amino acid sequence is completely different. And so before I started to study this, people didn't even know if the ATP synthase in Toxoplasma and other parasites related to Toxoplasma, like Plasmodium falciparum, that cause malaria, had a functional ATP synthase. They could find the rotor part that makes ATP, but didn't know how it was connected to the rest of the enzyme, didn't know what was happening in there. And so one of my papers, we show that there's actually, the subunits are there, it's just that when you use a program called BLAST to look for sequence homology, so the same amino acid sequences, you couldn't find anything because only the sequence is conserved, not only the structural of those proteins is conserved, not the sequence.
Wow. So one thing I wonder about, Toxo has all these extra ATP synthase subunits. Mm-hmm. And one of the things that goes along with it being such a successful parasite is that unlike a lot of parasites which have a host, am I correct in thinking it can infect all vertebrates?
Yes.
Which is crazy.
It can infect any warm-blooded animal, so not all vertebrates.
Okay.
But there's studies that show that if you put zebrafish at 37 degrees Celsius, Toxoplasma can infect.
Okay, that's crazy.
But in its normal life cycle, it's warm-blooded animals.
So if you had a warm-blooded dinosaur, Toxo could infect it.
Yeah, definitely.
Because we already mentioned Jurassic Park, so I thought I'd go
there.
We
did. We have to bring it back around.
Probably.
So evolution that's selected for all these subunits, does that have anything to do with the wide host range?
And can I also add in the wide cellular range? Because we're also talking about brain cells, muscles. Does that matter at all too?
Yeah. So we don't know. Definitely there's from an evolutionary point of view, there should be evolutionary pressure to keep those extra subunits in there, right? But we don't really know why. And it could be either, I don't know if it'll be regarding the host range. It might be the cell range, because when you think about the parasite, it will end up in a neuron or in a hepatocyte or in another type of cell that maybe the metabolism of the cell is completely different. So it might help with that. The other thing that we've discovered is that the ATP synthase also, it organizes in a very weird manner compared to other organisms. In yeast and mammals, it makes dimers. So an ATP synthase monomer will dimerize and make dimers, and the dimers then make a membrane curvature in the mitochondria, and that's what you call a cristae. And in Toxoplasma, the monomers will form dimers, and then 3 dimers will come together to form a hexamer. And so you have these hexameric arrangements that look like little pyramids at the top of this cristae in the mitochondria. And that shapes the mitochondria in a way that you don't have the morphology of the mitochondria. In yeast and mammals, you call it lamellar, they're lamellar cristae, but in Toxoplasma, they're tubular cristae.
So what I hear you saying is that if you do research on Toxo, you have a lifelong career because it does so many weird things. There'll be some new things to study all the time.
Definitely. That's what I'm here.
Well, and are there different strains of Toxo? I feel like we always talk about Toxo as Toxo, right? But we talk about malaria. We talk about all these different strains of malaria, right? That infect, have different ranges and things.
There's only one species, Toxoplasma gondii. But Toxoplasma gondii can be divided in different types.
Okay.
And these types are based on how fast they can kill mice in labs. So there's some strains that basically are, they're called type 1 strains. And those strains will be extremely virulent in mice. They will basically, one parasite is enough to kill a mouse in about 10 days. But then there's some other strains called type 2. And there's, at this point, I don't know how many types there are, but there's different types. But there's two that are commonly used in lab, type 1 and type 2. And the type 2 strain, instead of killing the mice, it will go into the cystogenic form and start making cysts and go dormant in the mice. But a lot of these strains, a lot of these types of parasites are also, there's two main types in Europe and in the US, type 1 and type 2. When you go to South America, there's dozens of types, specifically when you look in Brazil and the Amazon. And that's probably due to the fact that there, the definitive host of Toxoplasma are cats, but also felids. So any cat could be a jaguar, could be a house cat, could be as any type of felid living in the Amazon will have its own Toxoplasma type.
Oh, interesting.
Interesting. Yeah. I was just wondering if there were different types that you could get, a different type maybe is more likely to go into a certain place, and maybe then you could look at the different ATP synthases. Like, you know what I mean? You could isolate it to certain things.
So there's actually some types in South America that actually will cause disease even in immunocompetent patients, and they will cause ocular toxoplasmosis.
Oh, wow.
So those ones will go directly to your eye and then replicate in the eye, because the eye is one of those immunoprivileged sites in your body, and your immune system will be up and running. They will not care.
Okay, another silly question as the immunologist in the room. How does Toxo, quote unquote, know where the, protected sites are? Does it happen to get into these sites randomly? And then
that's a very good question.
It says this is good, let's stay here, right? Or are there mechanisms that have evolved with the organism?
I don't think we know at all.
Okay.
I think it might it might be a little bit of chance depending on how it gets. If it gets ingested, then it has to migrate into certain cells and then maybe ends up in a place that is more or less immunoprivileged. But I'll say it probably definitely senses, but I don't know how it will sense that.
Yeah, because everything, the gut immune system is pretty robust, and usually it can maintain things, right? So if you get ingested, it can kind of work locally to do something. But now you have an organism that's going to evade that and then get into places like the brain.
Yeah,
right?
Exactly.
It's kind of crazy.
If you eat raw or undercooked meat with cysts full of Toxoplasma, the pepsin in your stomach is what actually helps break down the cyst and then activates the parasites to then start wiggling around and then go somewhere else. So
let's touch a bit more about the cysts, because this is both fascinating and really creepy because you get infected, and I think, correct me if I'm wrong, but I think most people that are infected with Toxo, it's like at most a flu-like illness, they fever and they muscle aches for a few days. This is for someone that's immunocompetent. But then they're still infected for life.
Yes.
So the cysts, at some point, the immune system has controlled the infection. How does Toxo know to invade a cell and then turn into these slow-growing forms that we call bradyzoites?
Yes. So I don't think we have a clear answer to that. But what I know is basically the bradyzoites and the cysts, it's always under immune pressure that will decide to stop dividing and become dormant. And this dormancy is also something that we are trying to study because, and we, not only in my lab, but also other people in the field, this is a very hard stage of the parasite to study and also to treat. If you get Toxoplasma, sometimes you don't even get any symptoms. So if you're immunocompetent, but if you get then an infection and the parasite gets reactivated in your body, the only way to treat it is to kill the actively replicating parasites, which are called tachyzoids. So if they decide to also go back into the bradyzoid stage and insist, then you cannot really do anything about them, because most drugs that we use, and that's probably true for a lot of different pathogens, are targeting actively replicating pathogens, not the ones that are dormant, because the ones that are dormant, their metabolism is slowed down. The bradyzoids are still dividing to a lesser extent and slower than the tachyzoids, and the cyst can expand rupture and make new cysts. So we know that. It's not that one single cyst would stay with you forever. It's one single cyst might become tens of cysts, but they will not elicit any immune response and they will stay dormant.
So, bringing it back to ATP synthase, is there a difference in the ATP synthase of the bradyzoites, the slow-growing bradyzoites and the cyst? Or do we not know that?
That's one of the questions that I'm trying to answer in that grant that I submitted.
It seems like a pretty important question.
It does.
Because one of the things that we know about these parasites, and especially the metabolism of this dormant form, is that we don't know a lot. Most of the studies have used crude extracts of bradyzoites, have just mashed them up and then see, we have lactate dehydrogenase activity there, so they must be using lactate. But there's not been a lot of biochemistry done with them, only because it's hard to isolate a high number of them, also because it's hard to study them within the cyst. So my hypothesis, there's, at least bioinformatically speaking, when you look at expression levels of the different ATP synthase subunits between the different stages, there doesn't seem to be a very high difference between the known subunits, but that doesn't mean that there could be extra factors in the bradyzoites inhibiting or making something. It could also be that there's phosphorylations modifying the activity in bradyzoites versus tachyzoites. So that's also something that we're trying to understand. Basically,
knowing that this is a protozoan parasite is probably something ridiculously complicated.
Right.
Yeah.
Not like those little viruses. One of the goals of your lab we've read about is to uncover shared biological features and vulnerabilities across apicomplexans. Given how successful Toxo is, it might seem to have a few vulnerabilities. So what kind of vulnerabilities are you looking for?
One of the objectives of the lab is just to go back to the apicomplexans, and something we haven't mentioned is that not only Toxoplasma has extremely cool biology, I'm saying this in a non-biased way, but also Toxoplasma is a model apicomplexan. So the same way that people have used yeast or fruit flies to discover genes involving our biology and make breakthroughs regarding our biology, we use Toxoplasma as a model apicomplexan to understand the shared biology of all these parasites, because a lot of these parasites are very hard to study, are hard to manipulate genetically speaking, and so we use Toxoplasma for those. And for the vulnerabilities, I think one of the things that we're trying to do in the lab is try to uncover vulnerabilities regarding the ATP synthase, because the ATP synthase, I have not mentioned this, but it's essential for the parasite. So the parasite needs an ATP synthase, a functional ATP synthase to survive. Some organisms don't. For example, yeast, you can inhibit the ATP synthase, but they will do fine because they can also use other types of carbon sources to survive. Toxoplasma doesn't have that luxury. So it's a really essential enzyme. And based on the fact that you have all these divergent subunits and divergent features of the ATP synthase, we're trying to then find vulnerabilities, specifically try to target the ATP synthase with molecules.
Very cool. So your lab has another focus, which is communication between organelles, which I find really fascinating, because if we think back to the first cell biology course we all took, they taught us about mitochondria and endoplasmic reticulum and Golgi apparatus and lysosomes, but all as distinct separate things that had distinct functions, and they never mentioned. Maybe it's just because my cell biology class was many, many decades ago, but they never mentioned anything about them cooperating so much. Tell us a bit about how they communicate and why they communicate.
Yeah, so, I think my textbook was the same as yours in the manner that organelles are, defined entities defined by a membrane, and you can definitely see them working together, but maybe via vesicles. You'll have these vesicles that will cross the whole cell and be like, this is a vesicle from the mitochondria to the ER, and it has signaling molecules that will tell, you should go to the ER, and so on and so forth with other organelles. But in the last couple, I'd say maybe 10 years or so, there's actually this new field of structures called membrane contact sites. And these membrane contact sites are areas where the membranes of two organelles will be very, very close together, less than 30 nanometers. They will not fuse though, but that proximity enables the membranes to communicate and exchange a lot of things at a faster pace than vesicular trafficking. And also it's been uncovered that it's essential for the whole cellular homeostasis. A lot of these also proteins involved in membrane contact sites in humans are involved in diseases. So there's a whole subfield of cellular biology. And so one thing that the lab is trying to do, and I'm very proud of this because this is a thing that I scribbled down on a napkin on a break room like 10 years ago. And now I have a grant and people working on this, is how do the organelles in the parasite communicate with each other? Because this parasite, it's quite divergent from our cells and also has extra organelles that we don't have. So it has to deal with even more layers of complexity given the fact that it has extra organelles. And one of those organelles is called the apicoplast. And this apicoplast, it's a plant-like organelle, so it has plast like in plastid. The ancestors of these apicomplexans and of Toxoplasma engulfed red algae back in the days, 800 million years ago. And since then, it has become an integral part of them. And so how does this apicoplast communicate with the rest of the cell? We have no clue.
And is studying that also important for discovering something that you can target, given that we don't have an apicoplast?
Yeah, definitely.
Yeah.
That's another thing.
Yeah.
If phylum-specific organelles can also, they're already being targeted. There's antibiotics that you can use to kill it. The parasites that will target the prokaryotic machinery like of the apicoplast. But one of the main points of doing this type of research is that maybe we can also unravel new ways of targeting these essential organelles, because the apicoplast is so essential for the parasite.
Yeah. So how do you study the communication?
So to study communication is a very, it was quite complicated at the beginning. I think we're getting our hand on that. I'm hoping we'll publish something this year about this. But to study the communication, you first need to probe the membranes of the organelles and see what's happening at the membrane level. And to do that, other people have used in yeast and mammals. Basically, they create what's called a molecular handle. So imagine a protein that will be sticking at the surface of your organelle of choice. And then you can then use that handle to basically add on fluorescent proteins or other things to then label the whole surface of your organelle. And so those handles, of course, in yeast and mammals were known. In Toxoplasma, we had to start from scratch. Because a lot of the things that we in Toxoplasma is basically try to rig and adapt things that people have used in yeast and mammals. Half of the time it doesn't work, half of the time it's more luck and also, effort coming from us. But we do that, and so we have now molecular handles in 3 different organelles in Toxoplasma. In the mitochondrion, because they have a single mitochondrion, they don't have mitochondria. They have one single giant mitochondrion. In the apicoplast and also in the endoplasmic reticulum. And so we have these handles sticking out of each one of these organelles. And then we added to those handles a protein called turbo-ID. And turbo-ID, it's a biotin ligase, which means that when you add biotin to the cells, this biotin ligase would label with biotin anything that is around the protein, which is about a 30 nanometer radius.
So really close.
Really, really close. And so when you add biotin, we have the handles expressed in different strains. We don't have all handles at the same time. But what we do is, let's say we have the apicoplast handle, we add biotin, so anything that will be really close to the apicoplast membrane, any protein that will be really close will get labeled. And with that labeling, we can then do through biochemistry and mass spectrometry analysis, identify which proteins were next to the apicoplast and study them. And so what we did is we did this experiment 3 times with all these 3 different handles. And we have this, if you imagine a Venn diagram of proteins that will be overlapping between the 2 organelles. And that means that they're probably proteins that were on the surface of the other organelle as well. And then try to study their function. Because a lot of those candidates that we identify make sense, but a lot of them are just what I call hypothetical proteins. Because in Toxoplasma, the genome is 50% of the genome is annotated. The other 50% is a hypothetical protein, because it's a protein that doesn't have any known function compared to other organisms.
And this is a problem across the apicomplex and parasites.
Yes.
Which is really frustrating, because if you take any DNA from any mammal, even you discover a brand new mammal, and if you sequence it, you'll find that you can figure out what almost all the genes are.
Yes,
but
exactly.
When we do that in the apicomplexans, half the genes are assigned to a function, and the other half, the program says, I don't know.
Hypothetical protein.
Yes. So
Go figure it out.
Yeah.
Well, and that's also what I like, because again, when I see a hypothetical protein, we know where it is. It could be important, yes or no, and sometimes it's a gamble. But now they have more and more tools that we can be predicting more and more about these proteins, because it could be a hypothetical protein. It could also be essential for the parasite. That means that it's actually doing an important function.
Right.
It's more about how to study them rather than knowing exactly what it's doing.
So, Diego, when you're not studying ATP synthase or communication between organelles, what are you doing in your free time?
I play classical guitar. I've been playing classical guitar for, at this point, probably 20 years, I guess. So, because most of our family are musicians, and so one of my brothers is an actual professional classical guitarist.
Oh my goodness! Wow.
So I love to hear him play, and then I'm like, oh my god, I want to play that also. Can you send me, the sheets? And of course he sent me the sheets, but it takes me 5 months to learn what he learned in a week. So it's slow and tedious, but I like it. It keeps me busy and gives me a little bit of a getaway.
Well, if your brother ever comes to town and performs here, please let us know.
Yes, please.
Yeah,
please.
definitely. And I also do macro photography. So coming back to my love of bugs, I always like to take pictures of them, first with, crappy plastic cameras that were all blurry. Then we got cell phones and the pictures got better. And at some point, I really wanted to cross this definition and blurriness barrier that phones have. Still, some phones are getting way better, but they're not there yet. So I had a camera, and then I bought a macro lens, and so I started to take pictures. And I started basically during the pandemic, during lockdown. I found myself with a lot of free time. I was like, I think it's now or never. And being in lockdown, I can still go outside and look at wildlife. And so, yeah, I've been doing that now for a long time.
And I have to say, I found your site with your bug photos, and those are gorgeous. Okay, there's one thing you've left out of your spare time. Kim I explained this to her today. She didn't know about this, but every Monday when I get into work, on a desk just outside my office door, there's this cookie jar that gets filled up with amazing cookies. These are professional chef baker level cookies.
Well, and I do want to point out that when you pointed it out to me, that it is transparent, so you can can see the cookies,
which makes it even more difficult. And I think it's amazing you do this for the folks in your lab. It kills me because I see this and I know it's for the folks in your lab, and I've been very good about not taking it. But maybe we could cover it up so I can't see it.
Yeah, some aluminum foil, I guess.
But these are amazing looking.
Right.
Cookies.
So that's something else we can add to your free time, I think.
Yes.
Yeah, definitely. But it doesn't take too long, so that's why I don't really count it as a hobby. It's more like I realize that baking cookies is such a straightforward. If we're scientists, I'm following a protocol. And then
no, but you said you didn't like exact things. See, and baking is a very exact science.
Yeah.
That's why I don't like it, actually. I like cooking. Yeah. I just don't like baking.
Yeah, but for me, it's baking. I just have to. It's like making a PBS solution in the lab or any solution. Yeah. So,
our guest today has been Dr. Diego Huet. He's a faculty member in the Department of Pharmaceutical and Biomedical Sciences in the College of Pharmacy. Diego, thanks so much for being with us.
Thank you. It's been a pleasure.
So links to additional information about Dr. Huet's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched this season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemeth, for research, editing, scheduling, and keeping us on track.